Welcome back to the Bit-by-Bit Biostatistics Masterclass. We are officially beginning our deep dive into the intervention. This specific section marks a critical transition in your educational journey. We are starting Module 2, which comprehensively covers trials, causality, and usage laws. This module is built entirely upon the bit-by-bit pedagogy experimental core foundation. You are no longer standing on the sidelines merely observing populations like we did with case control and cohort designs. The paradigm completely shifts right now. You are now actively introducing a foreign variable into a biological system. You are the one manipulating the environment. The goal of this experimental design is to attempt to definitively and mathematically prove that your specific medical intervention is the absolute true cause of an observed clinical outcome. This active manipulation requires a much stronger and much more rigorous architectural frame than any basic observational study could ever provide. If you deeply memorize these specific mechanics and structural rules today, you will absolutely never miss an experimental design question on your board exams again. Let us initialize the trial protocols together and build your understanding piece by piece from the ground up. We are laying down the ultimate foundation for your future practice. The medical board examiners will heavily and relentlessly test your ability to spot subtle vulnerabilities in these clinical trials. If the experimental design is fundamentally flawed at its core, then absolutely no amount of perfect mathematical calculation can ever fix the final statistical result. We are actively building the absolute core of your medical evidence expertise right here and right now. The core foundation of this entire module is the experimental frame. Experimental studies in humans compare the therapeutic benefits of various active interventions. The overall quality and legal validity of this scientific frame relies heavily on three absolute pillars. It must be randomized, it must be controlled, and ideally it must be blinded. BIT says blinding is your primary defense against observer bias. If a doctor knows what the patient is getting, their subconscious expectations will inevitably leak into the data collection process. This psychological phenomenon can completely destroy the validity of an otherwise perfect experiment. Therefore, the frame must be randomized. Subjects are assigned to treatment or control groups by chance. This deliberate randomization eliminates selection bias at the source. It ensures that known and unknown confounding variables are evenly distributed between the two groups. The frame must also be controlled. Results are compared against a placebo or a standard of care. This control group provides the necessary baseline of truth. You cannot know if a drug works unless you know exactly what happens when the drug is not given. Finally, the frame must be blinded. Double blind means neither subject nor researcher knows the group. Triple blind keeps the analysts in the dark during the number crunching phase. This exact setup is the absolute bedrock of modern drug approval and scientific methodology worldwide. Master these three structural pillars, and you will completely master the foundational trial framework on your upcoming board exams. Every time you read a clinical trial abstract in a medical journal, you should immediately scan the methodology section for these three crucial concepts. If a study lacks randomization, you must question its entire selection process. If a study lacks a control group, you must heavily question its baseline measurements. If a study lacks blinding, you must strictly question the objectivity of its authors. Board examiners love to present you with a study that seems statistically significant, but secretly lacks one of these three pillars. They will ask you to identify the major flaw in the study design. By systematically checking for randomization, proper controls, and sufficient blinding, you create an impenetrable mental firewall against these tricky questions. We will now explore specific variations of this fundamental architecture. We will now examine a highly tested architectural variation known as crossover logic. This represents a robust, self-controlled architecture. Subjects receive a sequence of different treatments. Group A gets drug 1, then drug 2. Group B gets drug 2, then drug 1. BIT says crossover trials use fewer people because every patient is their own control. This is a massive statistical advantage because it completely eliminates interpersonal confounding variables like genetics or diet. But beware, because without a proper washout, you suffer from the carryover effect, which ruins the second phase. The washout period is absolutely critical. It is defined as a mandatory chronological break where no drug is given. It ensures the first drug is fully metabolized and cleared from the body. The provided image beautifully illustrates the crossover trial, complete with a screening phase, period 1, a crucial washout period, and period 2. If a board question describes patients switching their active therapies halfway through a formal clinical study, you must immediately search the text for the washout period. This ensures the final data is completely valid and untainted by residual pharmacology. The washout period must be mathematically calculated based on the specific half-life of the drug being studied. Generally, researchers wait for at least 5 half-lives to pass to ensure nearly complete elimination of the active compound. If the researchers fail to implement this chronological gap, the physiological effects observed in the second phase might simply be the lingering effects of the first drug. This carryover effect is the single most tested vulnerability of the crossover design. You must memorize this specific flaw. When you see a crossover design in a vignette, your brain should immediately trigger an alert to check the timeline. Did they wait long enough between interventions? If the answer is no, then the entire study is biologically invalid. This is how you outsmart the test writers and secure top percentiles. Now we must talk about the absolute law of intention to treat, or ITT. BIT says boards will try to tempt you. If 50 patients drop out because of side effects, you must still count them in their original treatment group for the final statistical analysis. The golden rule is once randomized, always analyzed. Patients are tracked in their original group regardless of what happens next. This preserves randomization benefits. It avoids bias from non-random non-compliance. In the real world, it reflects how patients actually take drugs in clinical practice. The image shows a cartoon of a classroom discussing clinical trials for pregnant women and children. This humorous illustration highlights the ethical and practical complexities of clinical testing. Real human beings are messy. They forget to take their pills. They experience adverse reactions and quit the study. They occasionally decide to take a different medication altogether. If researchers only analyze the perfect patients who followed all the rules, they create a highly artificial scenario that does not reflect real-world medical practice. Intention to treat solves this problem by forcing researchers to keep every single patient in their originally assigned bucket. This preserves the mathematical integrity of the initial randomization. It prevents researchers from artificially inflating their success rates by deleting non-compliant subjects. The boards will present you with scenarios where patients drop out of a surgical group and switch to a medical group. They will ask you how these patients should be handled in the final data analysis. The answer is always to keep them in their originally randomized group. This might seem counterintuitive because they did not actually receive the intended treatment. However, from a pure epidemiological standpoint, it is the only way to prevent catastrophic selection bias from ruining the entire experiment. Analyzing the failures brings us to the dangerous alternative methods researchers sometimes use. BIT says deleting or moving patients makes your data look cleaner, but it destroys the truth. Stick to the original assignments to prevent catastrophic structural bias in the trial. First is as-treated analysis. Subjects are analyzed according to the treatment they actually received, regardless of assignment. The risk here is destroyed randomization. Next is per-protocol analysis. Subjects who fail to complete the treatment exactly as assigned are excluded from the data pool. The risk here is massively increased bias. These alternative methods represent the dark side of statistical analysis. When a pharmaceutical company desperately wants a drug to look effective, they might be tempted to use a per-protocol analysis. By deleting all the patients who experienced side effects and dropped out, they artificially inflate the overall success rate of the compound. The boards will heavily test your ability to recognize these fraudulent analytical techniques. If a vignette explicitly states that researchers moved patients from the experimental group to the control group based on patient preference, you must immediately recognize this as an as-treated analysis. You must understand that this completely invalidates the initial randomization process. The two groups are no longer mathematically equivalent. The patients who actively choose to switch therapies are likely fundamentally different from the patients who remain compliant. By mixing these groups, the researchers introduce massive selection bias into the data. You must always advocate for the strict intention to treat protocol to maintain absolute scientific truth. Trial phases are formally divided into several chronological steps, starting with safe and work. BIT says the pivot point is the population. Phase 1 uses healthy people to check safety. Phase 2 uses sick people to check efficacy. You must prove safety before you prove it works. The mnemonic is "Can I swim?" Phase 1 asks, "Is it safe?" It uses a small number of healthy volunteers and focuses on toxicity and pharmacokinetics. Phase 2 asks, "Does it work?" It uses a moderate number of sick patients and focuses on clinical efficacy and optimal dosing. This progression is legally and ethically mandatory for all modern drug development. You cannot ethically administer an experimental chemical compound to a sick patient without first proving that it will not immediately harm a healthy human being. Phase 1 trials are often conducted on young healthy medical students or paid volunteers. Researchers start with microscopic doses and slowly escalate the concentration while monitoring liver enzymes, kidney function, and cardiac rhythms. They are hunting for the maximum tolerable limit. Once safety is definitively established, the drug graduates to Phase 2. Here the focus shifts to efficacy. Does the drug actually lower blood pressure? Does the drug actually shrink the tumor? Phase 2 utilizes a moderate number of patients who actually possess the target pathology. Researchers monitor clinical outcomes while continuing to evaluate short-term adverse effects. You must be able to read a vignette and instantly identify the current trial phase based entirely on the described patient population and the primary clinical goal. Trial phases continue their chronological progression with improve and market. We look at the specific phase, goal, population, and key comparison. Phase 3 looks for improvement. The population is a massive group of sick patients. The key comparison is versus standard of care, or a placebo. Phase 4 is for market surveillance. The population is the general public. It looks for rare or long-term side effects. Phase 3 is where the FDA grants approval, and Phase 4 is where rare side effects cause a global market recall. BIT says if a drug is pulled from shelves 3 years after release because of rare liver failure. That is Phase 4 in action. The public is the final permanent safety cohort. Phase 3 is the massive multi-million-dollar hurdle that pharmaceutical companies must clear to bring a drug to market. These trials involve thousands of patients across dozens of international hospitals. They must rigorously prove that the new experimental drug is statistically superior to the currently available standard of care. If it only works as well as an older, cheaper drug, the FDA might deny approval. Once the drug clears Phase 3, it enters the general market. However, the testing never truly stops. Phase 4 is perpetual post-marketing surveillance. When millions of diverse people begin taking a new medication, extremely rare side effects inevitably emerge. If a side effect only occurs in 1 out of every 10,000 patients, a Phase 3 trial of 3,000 patients might completely miss it. Phase 4 relies on vigilant physicians reporting these rare adverse events to national databases. This is exactly how dangerous drugs are eventually identified and permanently recalled from the market. Off-label applications take us entirely beyond the printed label. The image shows a pharmacy bag with various pill bottles and medications, illustrating this concept. Physicians use a drug for a condition, population, or dosage not specifically approved by the FDA. A classic board example is using tricyclic antidepressants to treat neuropathic chronic pain. This practice is legally permissible if scientific rationale exists and informed consent is obtained. Many medical students mistakenly believe that prescribing a medication off-label is illegal or constitutes medical malpractice. This is a massive misconception. The FDA strictly regulates pharmaceutical marketing and initial drug approval. However, the FDA does not regulate the independent practice of medicine. Once a drug is legally available on the market, a licensed physician can prescribe it for any logical medical purpose. Medical knowledge evolves much faster than bureaucratic FDA labels. It takes years and millions of dollars to get a new indication officially added to a drug label. Physicians simply cannot wait that long when a patient is suffering. Therefore, off-label prescribing is an absolutely essential tool in modern clinical practice. The boards will test your understanding of this concept by presenting a scenario where a doctor uses a familiar drug for an unusual purpose. They will ask if the doctor acted appropriately. As long as the doctor has a solid biological rationale and the patient understands the experimental nature of the treatment, the doctor is acting perfectly within the bounds of professional medical ethics. Proving the cause requires strict adherence to specific scientific criteria. These criteria build the massive wall of evidence required for establishing causality. BIT says one study in Japan is interesting. The exact same study in Brazil and Europe showing the exact same link is consistency. That is exactly how you prove a universal biological reality. Strength means large statistical associations are significantly more likely to be causal than small associations. Consistency means repeated observation of findings in different places, times, and populations. Specificity means the more specific the link between the cause and the effect, the stronger the evidence. The Bradford Hill criteria are a collection of philosophical and statistical guidelines used by epidemiologists to determine if an observed association is genuinely causal. In the messy real world, it is incredibly difficult to prove that chemical A directly causes disease B. There are always thousands of confounding variables. These criteria help scientists build a compelling logical argument. If a statistical association is incredibly strong, it is harder to dismiss it as a mere coincidence. If multiple independent researchers across the globe find the exact same association, it becomes nearly impossible to blame the results on localized laboratory errors. If a specific chemical exposure reliably produces one highly specific type of tumor, it strongly suggests a direct biological mechanism. You must memorize these specific definitions, because the boards will give you a clinical scenario and ask you to identify which exact Bradford Hill criterion is being demonstrated. Temporality absolutely comes first in the hierarchy of evidence. BIT says cross-sectional studies fail at causality because they measure both variables at once. If you do not know the exposure happened first, you have zero proof of causation. Cause mathematically leads to effect. The chronological rule dictates that the presumed cause must precede the effect by an expected amount of time. This is the only universally required criterion for establishing causality. Without a firm timeline, you only have a mathematical correlation, not true proof of causation. This is arguably the most critical concept in all of epidemiological science. You simply cannot get sick before you are physically exposed to the pathogen. This logical necessity is why retrospective case control studies and point-in-time cross-sectional studies are inherently limited. They can uncover fascinating statistical associations, but they constantly struggle with the timeline. If you find that depressed patients eat a large amount of junk food, you cannot immediately conclude that junk food causes depression. It is entirely possible that depression causes patients to seek out comforting junk food. This phenomenon is known as reverse causality. The only way to completely defeat reverse causality is to establish strict temporality. You must prove that the patients were eating junk food long before they ever developed the initial symptoms of depression. The board examiners will test this concept relentlessly. Whenever you are asked to identify the absolute most important factor for proving a causal link, you must always select the option that confirms the chronological timeline. Dose and mechanism comprehensively cover the concepts of biological gradient and scientific plausibility. BIT says if 5 cigarettes cause mild damage and 50 cause severe cancer, you have a solid biological gradient. This stepwise mathematical increase is a massive statistical indicator of active causality. The image beautifully shows the Bradford Hill criteria flowing logically from temporality all the way down to evidence. Biological gradient dictates that a greater effect is observed with greater exposure to the presumed cause. This is also widely known as the classic dose-response relationship. Plausibility requires that a conceivable biological mechanism exists to explain exactly why the cause logically produces the clinical effect. The dose-response relationship is an incredibly powerful tool for ruling out confounding variables. If an observed association is merely a random coincidence, it is highly unlikely that increasing the dose would perfectly correlate with increasing disease severity. However, if a chemical toxin is actively destroying liver cells, then logically a higher dose of that specific toxin should destroy more liver cells. This creates a beautiful linear graph that strongly supports a direct causal link. Plausibility brings the raw statistics back to human biology. Even if the math looks perfect, there must be a logical physiological reason for the connection. If a statistical model shows a strong correlation between wearing blue shirts and developing pancreatic cancer, scientists will correctly reject the finding because there is absolutely no plausible biological mechanism to explain it. You must always ensure the statistics align perfectly with known physiological principles. Vignette Skeleton 01 rigorously tests your understanding of crossover logic. The lead-in clearly states 50 patients receive an active migraine drug for 1 month, followed by 2 weeks of no medication, and then receive a placebo for 1 month. The critical pivot point highlights that the subjects are actively rotating through both groups, functionally serving as their own baseline controls. The discriminator explicitly asks what the precise scientific purpose of the 2-week gap is in this specific study design. The indisputable answer is a washout period, which physically prevents the carryover effect. This is exactly how you must approach every single question on your board exams. You must meticulously dissect the prompt into the lead-in, the pivot point, and the discriminator. The lead-in provides the raw narrative data. The pivot point is the hidden physiological or statistical concept the examiners are actively testing. The discriminator is the final question that forces you to apply your knowledge. In this specific vignette, the examiners are hiding the concept of the washout period behind a simple chronological timeline. By recognizing that the patients are switching therapies, you instantly identify the crossover design. By identifying the crossover design, you instantly know the major vulnerability is the carryover effect. By knowing the vulnerability, you instantly recognize that the 2-week gap is the required washout period. This systematic analytical process guarantees you will select the correct answer. Vignette Skeleton 02 deeply tests your ability to spot analysis failures. The lead-in states 1,000 patients are randomly assigned to experimental surgery. 50 terrified patients back out and demand the standard technique. Frustrated researchers analyze them in the standard technique group anyway. The vital pivot point is the researchers moved patients based entirely on the treatment they actually received, blatantly violating the original randomization. The discriminator directly asks what specific type of analysis was performed and what is the resulting statistical risk. The correct answer is as treated analysis, which carries a massively increased risk of selection bias. BIT says stick strictly to intention to treat. Even if they switch, keep them in their original group for the final math to permanently preserve the power of randomization. The board examiners absolutely love this specific scenario. because it tests your commitment to statistical purity over perceived clinical common sense. It feels clinically wrong to analyze a patient who received a medical pill as if they received a surgical operation. However, if you move that non-compliant patient, you completely destroy the mathematical equivalence of the two groups. The patients who refuse surgery are likely more anxious or perhaps more medically fragile than the patients who proceed. If you move them to the medical group, you are unfairly loading the medical group with highly anxious and fragile patients. This introduces massive confounding variables that will completely invalidate the entire multi-million-dollar trial. You must hold the line and demand intention to treat. Vignette Skeleton 03 rigorously tests your knowledge of trial phases. The lead-in explicitly states researchers administer a novel lupus antibody to 3,000 patients across 50 hospitals, comparing outcomes directly against standard steroids. The critical pivot point highlights that a massive group of sick patients is being compared directly head-to-head to the current standard of care. The discriminator asks, what exact phase of clinical trial this represents? The correct answer is Phase 3, which represents a massive improvement trial required for final FDA market approval. When you read a clinical vignette, you must constantly hunt for specific keywords that lock in a diagnosis or a study design. In this prompt, the keywords are 3,000 patients, 50 hospitals, and standard steroids. Phase 1 trials only use a few dozen healthy volunteers. Phase 2 trials only use a few hundred sick patients and rarely utilize active comparators. The sheer massive scale of this described operation, combined with the direct head-to-head comparison against an established active treatment, definitively points to a Phase 3 trial. The pharmaceutical company is spending hundreds of millions of dollars to definitively prove that their new antibody is mathematically and clinically superior to the old cheap steroids. If they succeed, they win FDA approval and massive profits. If they fail, the drug dies. This is the ultimate proving ground of modern medicine. Vignette Skeleton 04 tests your grasp of causality. The lead-in states an investigator finds a remarkably strong statistical correlation between high mineral levels and severe dementia, but notes the minerals were measured years after the clinical diagnosis occurred. The pivot point is the timeline is completely backward. The clinical outcome of dementia occurred long before the biological exposure to minerals was ever measured. The discriminator asks which core causality principle is most clearly violated in this prompt. The definitive answer is temporality, because the presumed cause must physically precede the effect. This vignette perfectly illustrates the extreme danger of reverse causality. A careless researcher might look at this strong statistical correlation and publish a sensational paper claiming that this rare mineral actively causes brain damage. The media would report it, and the public would panic. However, a well-trained physician like yourself would look at the methodology and instantly spot the fatal chronological flaw. Because the dementia was diagnosed first, it is highly probable that the progressive brain damage caused the patients to adopt strange eating habits or lose the ability to properly excrete the mineral. The disease caused the exposure, not the other way around. By rigorously applying the Bradford Hill criterion of temporality, you easily dismantle the flawed conclusion and protect your future patients from scientific misinformation. Module 02 is now officially complete. The intervention framework has been thoroughly mapped, and the laws of causality are now decoded. Synchronization of this critical data is successful. You now possess a deep understanding of experimental architecture. Intention to treat analysis strictly preserves randomization. Phase 3 explicitly compares novel drugs versus the established standard. Temporality reigns supreme as the first unbending law of causation. The required image sources are properly cited here. You have successfully navigated the most challenging conceptual leap in epidemiological science. You have moved from passive observation into the realm of active biological manipulation. You now understand how to build a clinical trial pillar by pillar, using randomization, control groups, and strict blinding protocols. You know how to process messy human data, using the unforgiving logic of intention to treat. You know how to track a novel pharmaceutical compound, from the preclinical animal lab, all the way to post-marketing public surveillance. Most importantly, you know how to logically and mathematically prove that an exposure actually causes a disease, using the Bradford Hill criteria. You are no longer just a student memorizing formulas. You are a highly capable critical thinker ready to evaluate complex medical literature and conquer the most difficult questions on your upcoming board examinations. We will build upon this massive foundation in the next module. This video utilized content from several sources, including toolbox.upati.eu, epomedicine.com, www.buzzrx.com, and joshualung.com.